Talking of Jason,
Good afternoon. Thanks very much, Sonia, and everybody else for inviting me today. I'm one of those researchers that this morning our keynote speaker said has not been a key researcher in ME, but he's actually moved from another field to try and give a contribution, applying methods which are being used in neurodegenerative disease, in diseases to actually try and understand a bit better what's going on in cognition and in the brain of patients with ME chronic fatigue syndrome. And we were very fortunate to get funding from Action for ME for a pilot study to look at cognition and functional connectivity, not in ME in general, but in a specific category of patients who have been diagnosed relatively recently, and in the condition where they have been exerted in the laboratory to experience post-exertional malaise. I don't need to give you much background because I'm sure that all the people in this room know much more than me. But what we know from the studies which are out there is that there is no definite understanding of what the brain abnormalities are and what the various symptoms that these brain abnormalities may give to contribute to the subjective experience of cognitive deficits in patients suffering from this condition. So what are the cognitive difficulties which are experienced by people who suffer from this condition? Well, there is a variety of them. As a neuropsychologist and a neuroscientist, I couldn't see a real specific pattern which could guide me anywhere. And there's also the sort of surveys which have been done. They seem to increase these symptoms when there is a high level of perceived fatigue. At the same time, when those very limited studies that looked at the brain of people who suffer from the condition, they found a variety of findings, very specific in my view, with regional cerebral blood flow deficit, but they could not be consistent with the cognitive studies. And the more recent application of sort of volumetric analysis have found very specific either frontal or occipital deficit. What this deficit in this area would have to do with the kind of deficits that subjectively people report was very hard to see. So we set off to do this pilot study to look at the pathophysiology behind these cognitive symptoms. First of all, to see whether the cognitive symptoms are already present in the very early stage of the disease. And we hypothesized that the significant abnormalities will be noted, especially when people will be stressed in the laboratory and they will be put in the condition of experiencing post-exertional malaise. So our design was as such. We had a T1 baseline, and I must say we have had a tremendous response for patients, which, due to limited funding, we had to turn away. We couldn't scan everybody. The scanning is very expensive, unfortunately. So we had a T1, we administered fatigue questionnaires, fMRI scanning, psychometric testing, and I will show you in a moment what we did. And then we brought back patients in the laboratory when they felt that they were very symptomatic and they were experiencing the exertion after the work they had done in the laboratory. And then our analysis stage was to look at the changes in fatigue level, look at the changes in resting state activity, and also have a comparison with what happens with controls who don't suffer from this condition. How did we induce post-exertional malaise in these patients? Well, we had a variety of tasks which increased in difficulty, and some of them required sustained and divided attention and high working memory load. Here are some examples. We used some fluency tests, but they were not the ordinary ones where you ask people to just name as many animals as possible. We included an executive component there by asking people to name animals with a specific characteristic, so they had to inhibit the spontaneous production that it was coming to their mind. Therefore, it is much more effort. You may try, if I say to name please name all the animals which have a horn, you will have lots of difficulties because lots of animals will be coming into your mind, but you have to inhibit those because that would not comply with the task. We also had, well, I've already mentioned this, but basically this is a description of what the verbal fluency and Stroop tests are. But we had also more difficult tasks where we were engaging working memory and sustained attention. And one of these was the PACE audio-serial addition test, where you're presented with numbers in sequence, and then each time you will very rapidly have to add up the numbers and push the relative button corresponding to the results. And this increases in speed. And of course, the perception is that you cannot keep up with the task. At the same time, another task we used to stress cognition was another working memory task known as the end-back task, where you have to press when you see a letter which is repeated with an interval of 1 or 2 or 3 blocks in between, or the letters in between. For fMRI scanning, we used a 3 Tesla machine, and we, as I said, we did scanning at baseline, at the time 1, and at time 2. And the analysis was done quantitatively, looking at voxel-based and also a functional connectivity. So we achieved the target of recruiting 10 patients and 10 controls, but not all of the patients, supporting what was said this morning in the keynote lecture, but also what Julia was saying earlier. Clear we have different phenotypes in this condition. So only 5 of our patients experienced post-cessation malaise within the range of a week. Others didn't experience anything, and we had to bring them back for a second session when 2 of those remaining 5 did actually experience this symptom. I'll show you some preliminary results because although we have analyzed all the cognitive and some of the imaging data, there is still quite a lot to do because it's quite complex analysis, and we completed the last subjects only a couple of weeks ago. But the results are actually, in my view, already revealing and interesting, although perhaps not exactly what everybody would expect. So our paradigm to exert fatigue in a post-cessation malaise was actually successful, and at least in 7 out of the 10 patients, fatigue increased and also the experience of pain increased. As you can see, controls didn't show any of this kind. When we looked at cognition and looked at patients versus control, not a great deal of differences, except for immediate memory, where a significant difference was found. For the rest of the battery we had for the evaluation of general cognition, the performance of these patients was exactly like that of controls. Likewise, when we did the retest with parallel forms of the tests after the post-cessation malaise. However, something which was interesting came out from the correlation analysis. When we looked at the correlation between performance on the tests on time 1 and time 2, what we did see was that the patients didn't seem to benefit from the fact of having been exposed to the testing on just a few days earlier, and it didn't show the usual practice effects that you would expect to see for somebody who has been exposed to the same kind of material. There is what is called in psychological terms a sort of priming effect. When you have already been exposed to a stimulus, you should be much quicker and do much better the second time. And indeed, for those tasks where this should have been more the case, in controls this priming effect was actually present, and you can see that highlighted in the box in this green circle. Now, what did we find in the imaging data? Well, we found something which is quite interesting in my view, and it goes a bit in line with what Jason was saying earlier, although maybe the connection may not be as obvious as one might think, or not immediate. What we found was significant abnormalities in the functional connectivity of the claustrum and some of the subcortical nuclei. But we also found a significant abnormality in the insula in these patients, and in some regions in the posterior parietal region, in the precuneus in particular. Well, you may say, all these nice blobs are fine, they're always very colorful, but what do they actually mean? Well, I think there is a nice parallel between what we see in the cognitive data, where we see the absence of practice effects, and possibly this reflects the subcortical nuclei dysfunction that we actually see in the functional connectivity data. What we know is that pathologies where there is a substantial dysfunction in the subcortical nuclei, in the basal ganglia especially, like in Parkinson's disease or Huntington disease, the effects of practice is actually not there, and priming is not present. So here we have a nice parallel. But something more interesting comes from the dysfunction which we found in the claustrum and in the insula. The claustrum is a key structure for multisensory and multimodal integration. And when we are in a condition that there is a high demand and tasks which have a very, very high load on our cognition, if we have a substantial dysfunction of a structure which is supposed to help multimodal integration, then this increases the demands of a task which is already perhaps high demanding, and lowers the threshold with coping with situations where there are demanding tasks. That might explain why in the situations or high demand, patients may experience a subjective experience of not being able to cope. At the same time, we have dysfunction in a key structure of the brain which deals with the sensation of pain and the evaluation of the intensity of pain. This might, in a way, lower the threshold in people who have this condition, and also signal wrongly about body perception of pain, and therefore the perception of the actual sensations that we experience is highlighted probably to a level which would not be normally. How do we explain the actual abnormalities in precuneus in the amygdala? Again, these are key areas for memory retrieval in one case, especially emotional memory retrieval in the second, but also in the emotion regulation and interpretation of emotion. And therefore, we have a nice parallel here between the subjective experience of patients and the brain abnormality that we see. One interesting thing which comes to this, which might be further investigated in terms of potential therapeutics, is that we are talking about circuits of structures, which is highly, especially the claustrum, which is highly reliant on acetylcholine transmission. And there is something which might be investigated there as a potential therapeutics. Now, where we go next? Well, first of all, we need to complete the behavioral analysis and perhaps try to relate the behavioral analysis and the lack of priming to the imaging data. And also, given that we have identified these regions as the key abnormalities, we would like to see how signal in that region relates to the rest of cognition, but also to the rest of signal within the brain. So do a sort of seed-based rather than whole brain analysis. And this is going to be completed in the next few weeks. This completes my talk, but I would like, first of all, to acknowledge the funding and thanks Action for ME for funding this study and sort of converting me to look at this disease, and also my collaborators on this grant, and thank you for your attention.